Jim Watkins and Annie Schofield from Cornell. We have data from Rick Barbiero and Barry Lesch and Beth Hinchy from US EPA. Luba Burlakova and Sasha Karatayev, Knut Miller come from SUNY Buffalo. My group and their group are the ones that now do the monitoring the Great Lakes National Program Office monitoring of the 5 Great Lakes. So that's why I'm here talking about Lake Huron when I typically more work on Lake Ontario. Ewan Reevy is also part of that from University of Minnesota Duluth, and we have a token Canadian, Todd Howell, that we managed to grab into the process in the end, which I'm very happy about. All right, so this is that push forward. So first, realize fish production requires production at lower trophic level. We know that is a clearly established structure. The more nutrients you have, the more fish you get. And if you want to get more information about this talk, see Tom Stewart's talk tomorrow. So what I want to do is just give you a status of Lake Huron and use these trophic level indicators. Spring total phosphorus, spring silica, water clarity, information on phytoplankton, zooplankton, mysids, which is near to my heart, diporrhea, and quagga mussels. So let's go through them. First, here's how we collect data. Collect data mostly from the Lake Guardian, so in the main basin of Lake Huron. Notice there's no points here at all. Georgian Bay, nothing here in the North Channel, and we don't go down to Saginaw Bay either. But the Canadians do have a nearshore sampling program, so that's where I got Todd Howell involved, and all of these sites are sampled every 5 years. So we can do some comparison from the nearshore from the Ontario Ministry of Environment, Conservation and Parks, right there? Yes. All right. I want to compare the 2 most recent reporting periods, which was Stephen told me to do. I thought he said 2005 to 2010, and not 2004 to 2010. So Stephen's right. Stephen's right. Where is Stephen? Stephen is right? Okay. This is right, because otherwise I have to redo all my statistics. And I'm also going to look at long-term trends and discuss a little bit the Cross Lakes. So here is some of the data graphs I'm going to show you. This is spring total phosphorus. Phosphorus is the limiting nutrient in Lake Huron. If we take this over time, move it over there, and then show you a little bit what I'm going to do. I'm going to look at the 2 time periods, so 2005 and 2005 to 2010 here in black, and 2011 to 2017 in either red or blue. So if it's blue, it means that there's a significant increase towards higher productivity of the lake. If it's red, it's a significant decline towards lower productivity of the lake. And here we see there is a significant increase in spring total phosphorus from this period to that period. There is, of course, clearly a significant decline in the long term. I'm also doing something called change points. I'm interested in if there's significant changes in the data series at a particular time. And here there is a significant decline in around 2004 and a significant increase again in 2010. That will come up because we're going to collect a lot of significant change points around that time period, 2003 to 2005. And I just wanted to discuss those a little bit in the end. In the Canadian nearshore, we see no change between 2009 and 2015 in spring total phosphorus. All right, silica. Silica is interesting because silica is what's taken up by diatom. So if silica is increasing, that suggests that we have less spring diatom blooms. So here we see an increase in the main basin. Now I put that red because that suggests lower productivity in the lake. There's a long-term increase in silica, again, suggesting lower productivity of the lake. Change points again in 2003, and actually a further increase in 2011. 11, and an increase in the Canadian nearshore. So all the silica indicate a continued oligotrophication of the lake through this time period. Water clarity. We measure that through SecuDisc. I have spring and summer data for you. Has also increased in this time period, so from the early to the recent time period, the water is clearer now in the spring. That's consistent with the higher silica concentrations. There's a long-term increase. There's a change point at about the same time as the other ones, like 2005, 2003, 2004, those time periods, and an increase in the Canadian nearshore. This one suggests increased, continuous decline in productivity of the lake. You can see that this way as well, so trying to put Lake Huron in perspective with the other 5 lakes. This is spring SecuDisc depth in 1983 to 1988. And just look at how these things change. Here is Huron. I have Huron in green. There's Superior. Huron has less water clarity than both Ontario and Michigan in this time period. Now, water clarity is higher in Huron than even in Lake Superior. So you have Michigan, Huron, Superior, all fairly similar. Ontario has gone up, and so has Eastern Lake Erie. That's in the spring. In the summer, we see no change in water clarity. We do have a long-term increase. We have an early change point, not fitting with the other ones. But in the Canadian nearshore, there's actually a decrease in water clarity between 2009 and 2015. So summer water clarity has not changed as much as it has in the spring. So let's now go to the phytoplankton. So phytoplankton have various ways of measure. One way we've done it is with satellites. Barry Lesht, working with Rick and us, has put in looking at all the satellite data and come up with various, both for Georgian Bay and from the main lake. So this is spring chlorophyll in May, and there's been no change in the main basin. You can see that. Has there been no change in Georgian Bay either, because we have all that variability, we can't detect it. But we have seen a decrease in chlorophyll in the main basin, and this one should have been blue. I forgot that, look at that. So this is a blue arrow towards increased chlorophyll in Georgian Bay. Change point again around 2005, and no change in the Canadian nearshore areas. Just, hopefully we can put it all together in the end, right? So we do, Ewan Reevy up in Minnesota does phytoplankton biovolume measurements. So he takes samples, measure phytoplankton cells, and calculate how much biomass or biovolume you have. For them, we only have data back to 2001, and there is this rather fast decline here, both in the spring and in the summer around 2002, 2003, but we have very few data points on the other side, so it's hard to detect. We do see a decline in the spring, which is consistent with the increase in silica and increase in water clarity. We see no change in the summer, which is also consistent with no change in water clarity in the summertime. I do love it when my indicators are consistent with each other. That's nice. Water clarity, what we talked about here was surface waters, the epilimnetic values. We also have chlorophyll deeper down under the thermocline. And when you get lower productivity lakes, you often get a peak in chlorophyll below the thermocline because there's enough light there for phytoplankton to grow. However, we have looked at these now and we find that the chlorophyll levels are higher. This is the surface layer, that's the deep chlorophyll layer. They are higher in the up here, both in Huron and Michigan, but they are going down. So we don't see a deep chlorophyll layer compensation for the decreases in the epilimnatic chlorophyll. All right, moving on to zooplankton. So this is some of the ones we're going to look at. This is the Limnocalanus, a big omnivorous copepod, cold water copepod. These are other calanoid or diaptomid copepods. We put them blue. They're both blue in my eyes. One is a little different blue colors. Cyclopoid copepods, Daphnia in red, and mostly Bosmina in this purple color. So some data on summer zooplankton. There has been no change in summer zooplankton over this time period. So this is consistent with this no change in water clarity, no change in chlorophyll and biomass of phytoplankton in the summertime. But we have, let's see what comes after that. We do have a long-term decline, and certainly a very dramatic change here in 2003 when we lost the Daphnia. The other species are still there. I like to note that my counters really worked hard to get you a 2018 number in the graph. I think this is the first time we presented a Lake Committee meeting from Glencoe where we actually managed to get the year of last year's data in. Thank you. So if you compare that with other lakes, Ontario, Michigan, Huron, and Superior in 1997 to 2002, this is what we have in 2013 to 2017. Pretty strong decline in Lake Huron. Very similar now between Michigan, Huron, and Superior, and only a little bit higher in Ontario. Whereas here there was more in Huron than in the other lakes. Your lake is fascinating. What about mysis? So in some of the lakes around, in Lake Ontario, for example, we can have mysid biomass being 20 to 30% of the total zooplankton crustacean biomass. And that's not included in any of these numbers that I showed you so far. We have a spring decline and a summer no change in the data we have since 2006, but that's only those two periods we just compared. I don't have this kind of mysid-specific data back in time. I only have that if we use the zooplankton net data, which is questionable. But if you do that, you have a decline, long-term decline, and you have that change point around 2005. Mysids, comparing with the other lakes, is lower in Huron than in Michigan, Ontario, or Superior. It's higher than Erie, you have that, but it's lower than the other ones. What about these benthic invertebrates? You've seen these pictures before. The Nalepas pictures when they are plenty in 2002, declining as you go through it in 2012. And actually we did find, this is Karatayev and Mailer's data, did find some still up there in the northern part of Lake Huron. But certainly substantially less than earlier. If one looks at the annual data series, which is not as spatially extensive, just I think it's 14 sites. Here is shallow diporrhea, clear decline, early, deeper, decline. But since, let me go back here once. Since that time period though, we have not seen further decline in deep water diporrhea. Shallow water diporrhea are pretty much gone. So there has been a long-term decline in both of them. Change point, again, 2004. Quagga mussels. There's no zebra mussels in Lake Huron anymore. Quagga mussels. They have been increasing and now covering all of the main basin. This is one of the few lakes where quagga mussels have continued to increase lake-wide. They started to decline in the shallow waters, but have continued to increase as a whole lake, especially in deeper water. So I'm actually going to jump first to this one. Lower trophic levels long term. The lake is getting more oligotrophic. You knew that. You've seen it in all the other indicators. And there is no indicator at all that is blue here. So all of them shows a decrease in phosphorus, increase in spring silica, increase in water clarity. Chlorophyll is decreasing. Deep chlorophyll is also decreasing, which surprised me some. The copepods, the Daphnia, everything is increasing except quagga mussels. So quagga mussels are increasing. Now I want to go this way. But if you just look at the two last time periods, so 2005 to 2010 and 2011 to 2017, the picture is a little bit different. We do have an increase in total phosphorus, so it seems to be stabilizing some. Spring silica is increasing, so that shows less diatoms in the spring, but the summer there is no change in water clarity. The summer there is no further change in phytoplankton biovolume and no change in chlorophyll. Deep chlorophyll was decreasing. There's a small increase in some of the zooplankton copepods. Mysis no change in the summer. Diporrhea no change in the deep waters. Quagga mussels are increasing. So it has stabilized. It's not going down as much as it did before, and the decline is primarily in the spring. Note that quagga mussels are increasing mostly in the deep waters. They have, as we know from the studies in Lake Michigan, they have mostly access to the water column in the spring if they are in deep waters. So I think that is the reason why we still see declines in all these indicators of spring phytoplankton, spring chlorophyll, deep chlorophyll layers, decrease in diatom blooms, is due to this increase in quagga mussels, not to further declines in spring in phosphorus. And in the summer, quagga mussels in the deep water should have very little effect on the productivity of the lake. It should be mostly run by how much nutrients there are there, because they don't have access to that water column. And that's what we see. So, you do have long-term oligotrophication of this lake. It will not produce as much fish as it used to. Spring values, spring oligotrophication has continued in this since 2005 and is further lower productivity in the spring, but not much change in the summertime when you look at these two recent time periods. And these kind of changes overall, I think, is consistent with the increase in lake-wide quagga mussels and the effect of those being most important in the spring. So what happened in 2003 and 2004? Why did Daphnia decline? Well, there is increased water clarity, but that could have caused an increase in mortality of these Daphnia, but we don't think that's the case because the alewife disappeared. So there's not that much fish predation left. I think it has to do both with quantity and quality of phytoplankton. Not just the quantity of them, but also the quality. And we do can measure quality of phytoplankton by taking the ratio between carbon and phosphorus. And the higher that ratio is, the more empty calories, so to speak, you have in carbon and less nutrients you have in the food that they eat. And we know that in Lake Superior, the C to P ratio is something around 250, 280, which is a level which affects Daphnia reproduction. And in Lake Huron, I've seen this creeping up over this time period. I should have plotted that to now being at the levels which limits Daphnia reproduction. So I think it's both decreased quantity and decreased quality of the phytoplankton, which has to do with these, we can measure with the ratio between carbon and phosphorus. And finally, these results were mostly from the main lake here. I told you a little bit about the nearshoring, the Canadian side. I have no data from the North Channel to talk about, and I did not include data from Saginaw Bay just because I didn't have access to them when I wrote that chapter, which is unfortunate. It's still a productive part of the lake. It produces maybe 30% of the total nutrients that come down into this part of the lake. So I should have included that, but I didn't. Thank you.